Hi everyone, Ryan Duffy here from Precision Microbes. I'm here in TUS. I'm joined with Kian Ahearn here, who's done a project on silage, on using our silage additive for silage fermentation. So Kian, thanks very much for having us.
Thank you.
Just tell me a wee bit about yourself there, Kian. Like, you're obviously a student here.
Yeah, I'm a fourth year microbiology student now in TUS, just about to graduate.
Very good.
And some experience I have, I went on work placement in the clinical microbiology lab in Mullingar Hospital. So I did 5 months unpaid, and then they kept me on for the summer then to pay me. And some modules I've done in college that relate to this are environmental, I suppose food microbiology as well. And yeah, I really enjoyed doing this project, and there was some great results in it as well.
Yeah, it's always interesting to kind of get into practical applications of microbiology, because like obviously it's a massively underexplored component as to what good things bacteria can actually do for us.
Yeah.
So what are your ultimate findings here by using the silage additive?
So in the treatment groups then, we've seen a massive rise in the levels, the CFU per gram of silage of the lactic acid bacteria. In the control group, it went from 2.935 times 10 to the 7 all the way up to 3.925 times 10 to the 8. So there was a big increase, and it was able to establish dominance there.
Yes.
So the total viable bacteria then, excluding the lactic acid bacteria, it went from 1.835 times 10 to the 9 in the control group, all the way down to 8.317 times 10 to the 6 in the treatment group 2. So there's a big, huge change in population.
And those would be the bad sort of bugs that we
kind of undesirable, like you want as much lactic acid bacteria as possible, because they are what you want to do fermentation.
Yes.
They produce the lactic acid to do good preservation.
And I see you have 98% of the bugs that are actually present there are lactic acid bacteria.
Yeah, in treatment group 2, they made up on average 98% across all the groups.
Very good. So a clear predominance, which is exactly what we want within our grass silage.
Yeah.
So what's the ramification of actually having those lactic acid bacteria there in such high concentrations?
Well, just one thing I'd like to say is the lactic acid bacteria can be categorized into 2 main groups. There's heterofermentative species, which break down carbohydrates into lactic acid, acetic acid, ethanol, and CO2. And this is slower at decreasing the pH, and it's not as good at fermentation. But Enterococcus faecium is a homofermentative species, and it breaks down carbohydrates into mainly lactic acid, which makes up 90% of the end products via glycolysis, and it is a quicker acidification and an overall better fermentation.
Yeah. And did you see that here by having that predominance of lactic acid bacteria?
Because just here in the lactic acid contents, it went all the way up to 378 micrograms per litre detected in treatment group 2, and in the control group, with the test strips we used, it was unable to detect any at all in the control group with no added lactic acid bacteria.
Okay, so pretty much the higher amount of silage additives put in, the higher level of lactic acid bacteria, the higher level of lactic acid, and therefore you're aiming for better nutrient preservation.
Yeah.
Did you see that in this study?
Yeah, we did, because in the control group here, the ammonia levels, so ammonia in silage is a sign of protein breakdown, so that's a bad thing. And in the treatment groups, it went down. So in the control group, it was 30 micrograms per litre detected, went down to 18.75 in treatment group 1, and in treatment group 2, it went down to 10 micrograms per litre. So that means there's a better preservation of the proteins, and of course that's better for growth in animals and reproduction and milk production.
For sure. No, that's exactly what we're aiming for. Like, we can get the protein into the animals, no doubt we'll get far better performance and productivity. So it's very interesting now with all of these components as to what you've actually done here with your control and the 2 different dose rates of the silage additive, and obviously the results. Like, we've got far better lactic acid concentrations, far bigger predominance of lactic acid bacteria, and most importantly, as you said, less ammonia and nitrogen.
Yes.
Massively important.
Yeah.
So no, listen, Kian, really appreciate it. Very interesting stuff. We'll definitely be meeting this project again over time. But yeah, no, thank you very much.
Thank you.